Microbiology, the scientific study of microorganisms, has a rich and fascinating history spanning centuries of human curiosity. Microbiology is defined as the study of organisms too small to be seen with the naked eye, including bacteria, fungi, viruses, archaea, algae, and protozoa. Microbiology emerged as a scientific discipline through several distinct phases, starting with early observations and progressing through scientific debates, experimental proofs, and finally becoming the modern field we know today. The development of microbiology rested on three fundamental pillars: careful observation, rigorous experimentation, and vigorous scientific debate. Early microbiologists challenged long-held beliefs, such as spontaneous generation, replacing them with evidence-based principles like biogenesis, the understanding that life comes from pre-existing life. These early scientific pioneers established methods and principles that remain foundational to microbiology today, creating a legacy that continues to influence modern scientific understanding. As we continue our exploration of microbiology's history, we'll examine the pioneering observations that launched this fascinating field. A central debate in early microbiology was the theory of spontaneous generation, also known as abiogenesis, which proposed that living organisms could arise from non-living matter. For centuries, people believed that living organisms could arise spontaneously from non-living materials. Common examples included mice appearing from stored grain, maggots emerging from rotting meat, frogs forming from mud, and microbes developing in broth. In 1668, Francesco Redi conducted a groundbreaking experiment to test the spontaneous generation of maggots from rotting meat. He placed meat samples in two sets of jars, one set left open and another set covered with gauze that allowed air to enter but prevented flies from reaching the meat. Redi observed that maggots only appeared in the open containers where flies could land and lay eggs. The covered jars, despite containing the same rotting meat, did not produce maggots. This experiment provided evidence against the spontaneous generation of maggots from meat. In 1768, Lazzaro Spallanzani took the investigation further by focusing on microorganisms in broth. He placed nutrient broth in flasks, with some sealed and others left open. Then he boiled all the flasks to kill any existing microorganisms. Spallanzani observed that the sealed flasks remained clear with no signs of microbial growth, while the open flasks quickly became cloudy with microorganisms. This experiment suggested that microbes didn't appear spontaneously, but entered from the air when the flask was unsealed. These experiments by Redi and Spallanzani were crucial challenges to the theory of spontaneous generation. They provided empirical evidence that living organisms don't arise spontaneously from non-living matter, but instead come from pre-existing life forms. Their work laid the groundwork for Louis Pasteur, who would later deliver the definitive blow to spontaneous generation theory. Louis Pasteur, a French chemist and microbiologist, definitively resolved one of science's most controversial debates in 1861. For centuries, scientists had debated whether living organisms could spontaneously generate from non-living matter, or if life only arose from existing life. Pasteur designed an ingenious experiment using specially designed flasks to test these competing theories. In both flasks, Pasteur added nutrient broth, then boiled it to kill any existing microorganisms. The swan-necked flask's curved design would allow air to enter but trap dust particles and microbes in the bend. After a few days, the broth in the standard flask became cloudy with microbial growth. However, the broth in the swan-necked flask remained clear indefinitely. Pasteur's elegant experiment conclusively demonstrated that microorganisms do not spontaneously generate from non-living matter. Instead, they enter from the environment, specifically from microbe-laden air. This established the fundamental principle of biogenesis, that life only comes from pre-existing life. Pasteur himself recognized the significance of his work, stating, "Never will the doctrine of spontaneous generation recover from the mortal blow struck by this simple experiment." His work forever changed our understanding of the origins of life and established the foundation for sterile laboratory techniques still used today. Louis Pasteur's work on fermentation would lead to one of the most revolutionary concepts in medicine. Before Pasteur's work, fermentation was thought to be a purely chemical process. Pasteur discovered that microorganisms were responsible for this process. In 1859, Pasteur demonstrated that fermentation was caused by living microorganisms, not just chemical reactions. This discovery led Pasteur to a revolutionary insight about the cause of diseases. In 1865, Pasteur proposed the germ theory of disease, the revolutionary concept that specific microorganisms cause specific infectious diseases. This was a profound paradigm shift in medicine, changing how humanity understood the cause of disease. Before Pasteur, diseases were often attributed to miasma, bad air, or seen as spontaneous occurrences or even divine punishment. Pasteur's germ theory changed this view completely. Now, diseases could be understood as infections by specific microorganisms, potentially preventable and treatable. Pasteur's further work on anthrax in 1876 helped validate his theory, as he identified the bacterium responsible for this deadly disease. The germ theory of disease became the foundation of modern medicine, revolutionizing hygiene practices, surgical techniques, and disease control methods that continue to save countless lives today. The period from 1880 to 1900 is considered the golden age of microbiology, an era of unprecedented scientific discovery. During these 2 decades, the foundation of modern microbiology was established through rigorous scientific methods and breakthrough discoveries. Students of Louis Pasteur and Robert Koch rapidly advanced the field by identifying numerous disease-causing bacteria and developing new laboratory techniques. Several pioneering scientists made remarkable contributions during this golden age. Emile Roux worked with Alexandre Yersin to develop the diphtheria antitoxin in 1883, saving countless lives. In 1887, Richard Petrie invented the shallow-covered dish that revolutionized bacterial culture techniques. Shibasaburo Kitasato and Emil von Behring discovered tetanus and diphtheria antitoxins in 1890, establishing the field of serum therapy. Dmitry Ivanovsky was the first to recognize filterable agents, later known as viruses, in 1892. Martinus Beijerinck confirmed the viral nature of tobacco mosaic disease in 1898, coining the term virus, from Latin for poison. The identification of disease-causing bacteria accelerated dramatically during this golden age. Major bacterial pathogens identified during this period included the causative agents of tuberculosis, cholera, diphtheria, tetanus, and plague, fundamentally changing our understanding of infectious diseases. Revolutionary techniques developed during the Golden Age transformed how scientists studied microorganisms. The Petri dish, agar plating methods, and bacterial staining techniques are still fundamental to microbiology laboratories today. The Golden Age saw an exponential growth in microbiological knowledge, with profound impacts on medicine and public health. By 1900, scientists had identified nearly 100 bacterial pathogens compared to just a handful known in 1880. This rapid expansion of knowledge directly led to improved disease prevention, diagnosis, and treatment methods that saved millions of lives. The Golden Age of microbiology established the scientific framework for understanding microorganisms that continues to evolve today. Several key discoveries revolutionized the field of microbiology. In 1796, Edward Jenner developed vaccination, using material from cowpox to provide immunity against smallpox. This revolutionary approach led to the first scientific immunization method, and eventually to the global eradication of smallpox, saving countless lives. Moving forward in time, another remarkable discovery changed medicine forever. In 1928, Alexander Fleming discovered penicillin when he noticed that a mold contaminating his bacterial cultures created a bacteria-free zone. This accidental discovery led to the first antibiotic, saving millions of lives and transforming modern medicine. Earlier, scientists made another crucial discovery that expanded our understanding of infectious agents. In 1892, Dmitry Iwanowski discovered that the tobacco mosaic disease agent could pass through filters that retain bacteria. 6 years later, in 1898, Martinus Beijerinck expanded on this work, showing the infectious agent could diffuse through agar gel and coined the term virus. These three key discoveries, vaccination, penicillin, and viruses, revolutionized our understanding of infectious diseases and transformed medical science.